Good morning to all the CUET-PG Botany aspirants. So, here we are to discuss the 60 days roadmap to crack the exam gunshot. Okay. So, in this session we are going to discuss CUET-PG Botany 60 days strategic plan. How can we implement this? Let us go through. From day 1, that is, we are going to start. Okay. So, here we see day 1 we are going to exercise microbiology, bacteria, its structure, nutrition and reproduction. Microbiology 1. In first session we are going to exercise bacteria, its structure, nutrition and reproduction. Then we will go for an objective practice. Clear, right? And then if we see on the other day, on the second day it will be the objective practice. Third day we will look on to microbiology 2, wherein we will be highlighting the bacterial diseases, angular leaf spot of cotton, bacterial leaf blight disease and economic importance of bacteria. Then we will move on to virology 1 and 2, where on 4th and 5th day we will be exercising clearly on virus structure and replication, and then about clear description on tobacco mosaic virus and rice turgor disease. In replication, of course, we will be highlighting lytic cycle and lysogenic cycle as how we have discussed in the syllabus. As such, in 4 days, we will be finishing off microbiology. And then we will move on to phycology. Phycology, we will be taking 3 sessions. Algae 1, where we will be focusing on general characteristics, classification and reproduction, and algae 2, that is phycology, we will be looking into chlorophyceae and phaeophyceae, and then we will be focusing on rhodophyceae. So, this is the first week session or planner. Is this clear? Any doubt anywhere, ma? Yeah. Very good morning, ma'am. Just continue with the class. Yeah. So, next if you see here, just a second, yeah. So, and then on 9th day again we will be moving on to objective practice session 2. In every objective practice, we will be focusing on the questionnaire onto half the syllabus is completed. Like at least 50 to 60 questionnaire we will be practicing of your CUET-PG Botany. Fine, right? And then mycology, its classification, structure and reproduction. Most importantly, asexual sexual spores are the targeted hotspots from mycology all the time. And then important fungal genera like Albuca and Mucor from Zygomycotina, and genera 2, Penicillium from Ascomycotina, Puccinia from Basidiomycotina. We will be focusing on the general characteristics and type studies as such. And then if you see phytopathology, where plant pathogens like fungal pathogens, bacterial pathogens, viral pathogens will be discussed in detail, and we will also look into the execution of disease control practices, which is most important. Then we will go on through lichens, its ecological and economic importance, and then objective practice session 3. This will be the second week sessions, okay, along with the objective practice, and each objective practice will be exercising around 50 to 60 questions by drilling up the concept, okay. And then yeah, archegoniate will be starting from 17th, day 17th, where in archegoniate 1 will be looking up to Marchantia, Anthoceras, its structure and life cycle in detail. Then Bryophytes archegoniate 2, where we will be focusing on Polytrichum, its structure and life processes. And then we will be moving on to archegoniate, that is Pteridophytes, under which Lycopodium, Equisitum, Marsilia will be covered. Most importantly, targeted concept is stellar evolution and heterospory. Okay, Pteridophytes, that is stellar evolution and heterospory we will be looking into. And then gymnosperms archegoniate like Pinus and Neatum. This will be covering up. And then objective practice session 4. Here we go. Gymnosperms archegoniate 2, reproduction and its economic importance, followed by paleobotany, fossils, fossilization, geological time scale. And then plant anatomy will be looking into meristems, tissue systems, and leaf anatomy, root anatomy, stem anatomy, comparative analysis between dicot, monocot, root, stem and leaf. As such, we will be covering up. And then anomalous secondary growth, where normal and anomalous secondary growth taking different timbers into consideration. Then on 28th, we will be looking into day 28, we will be focusing on angiosperm embryology, where we will be highlighting the contents like anther, ovule, gametophytes and pollination. Then angiosperm embryology 2, session 2, where we will be focusing on fertilization, embryo and endosperm development. That is double fertilization and post fertilization events we are going to target, followed by objective practice session 5. Fine, right ma? So till half of the days, that till 30th day we have finished 5 objective practices and covered from microbiology, phycology, mycology and anatomy, morphology, followed by angiosperm embryology till post fertilization events. Clear till here the concepts? PG life sciences will be better for you beta. Yeah, if you have post graduation in pharmacy, then you can go for PG life sciences. Okay, and the applied biology concepts over here, now you will see in the next 30 days where we will be covering on cell biology, genetics, small bio, that will be there for life sciences as well. Okay, that will be useful for life sciences aspirants as well. Fine, right? Now, here we will move on. Yeah, from 31st we will be starting up the applied biology concepts. Okay, applied biology concepts where we will be focusing on cell biology 1 to 31st day and 32nd, followed by chromosome structure and special chromosomes or giant chromosomes like polytenic chromosomes and Lamb-Rush chromosomes are very important over here. And then followed by chromosome shapes and then cell division, both mitosis, meiosis and cell cycle regulation, highlighting the checkpoints, cyclins and CDKs we have to learn. Then biomolecules, most important biomolecules, like we will be looking up to the structure, biological functions and biosynthetic metabolism of carbohydrates, lipids, proteins and the nucleic acids, followed by objective practice session 6. Till here it is clear, right ma? Till here it is clear, right? And then this is useful for you also ma. From date 31 even you can join. And here next session from 38 we will be looking into enzymes from biomolecules, where in enzymes we will be looking into enzyme nomenclature, enzyme classification, followed by cofactors, enzyme mechanism of action, where we will be looking into lock and key hypothesis, Koshland model. As such, complete enzymology and finally factors affecting the enzyme activity also we will learn, right ma? pH, how pH is affecting the enzyme activity, how temperature is affecting the activation energy. Completely we will look into enzymology 1 and 2 on 38th and 39th day, followed by 40th day we will be looking into genetics, Mendelianism, principles of inheritance, chromosomal theory of inheritance, and then followed by advanced genetics. In advanced genetics we will look into those exceptions of Mendelian's principles like incomplete dominance, codominance, multiplalism, pleiotropy, followed by polygenic inheritance, and we will also learn about non-allelic interactions. Non-allelic interactions include what? Epistasis. Epistasis is the major thing we are going to learn here. Fine, right? Next, 42nd, we will be learn on 42nd day linkage and genome mapping. Linkage, crossing over, recombination, gene mapping we will be learning. Fine, right? And here along with epistasis we will learn supplementary gene interactions, inhibitory gene interactions, complementary gene interactions, everything we are going to learn. Around 6 to 7 types of non-allelic interactions will be there. We will be covering up them. Followed by mutations, most important segment in malbio or genetics. Mutations, like what are different types of mutations? Gene mutations, chromosomal mutations, again chromosomal mutations based on the difference in chromosome number or on the structure. As such, we will be having a detailed flowchart and complete types of mutations we will be exercising, and looking into physical and chemical mutagens, whichever are causing mutations are addressed as what? Mutagens. Physical and chemical mutagens also we will go through. Then on 44th day, objective practice session 7. Fine, right? So till 44th day we have finished up what? 7 objective practice session. First 30 days we are focusing on core botany chapters. Next 30 days we will be exercising the applied biology chapters, which is useful for the other stream as well. Like whoever are going to write for life sciences, whoever are going to write for zoology, for them also these concepts will be useful. Cell biology, genetics, fine, right? Mol bio, biochemistry, this and all will be useful for them as well. Now, yeah, on 45th day, before going to this, I would like to address one more thing. I did get you, ma. Please type it once again. Ma'am, what is comet essay? Go through this. Yeah, enough. This session is only concerned for CUET, ma. Yeah. So now, 45th question, if you see, molecular biology in this, starting from the structure of DNA and RNA, its functions, types of DNA polymerases, types of RNA polymerases, followed by replication, transcription, translation and gene expression. Complete contents we are going to cover in 2 days. And then we will start with plant ecology, where we will be highlighting ecosystem components, energy flow, food chains, food webs and ecological adaptations like hydrophytes, xerophytes and mesophytes. And then plant succession includes what? 2 types of plant successions we have to learn, hydroseed and xeroseed. Then plant physiology will be starting from day 49, like transport in plants, and then we will be looking into mineral nutrition like macro and micronutrients, followed by deficiency symptoms. Clear, right? And then we will have objective practice session 8. Till here everything is clear, right, ma? Now we will see the plan of action for last 10 days. Transpiration, mechanism of stomatal opening and closure, followed by photosynthesis, dark reactions or light independent reactions, where we will be learning on C3, C4, CAM pathways and photophosphorylation as well, that is light reactions, cyclic, non-cyclic photophosphorylations, followed by dark reactions, and we will also look into C2 cycle, that is photorespiration. Then respiration again. These cycles are important for non-core botany students as well, whoever are writing CUET-PG. So here this includes glycolysis, Krebs cycle, oxidative phosphorylation, electron transport system. Then nitrogen metabolism, biological nitrogen fixation along with the mechanism. And then 2 days we will be focusing completely on plant growth regulators only. Okay, plant growth promoters, plant growth inhibitors, where for each hormone we will be learning, each phytohormone we will be learning its mode of discovery, mode of action, what are its precursors, okay, what are its derivatives, fine, right, what are its biosynthetic pathways, what is its application, what is its biological use, everything we will be learning for each plant growth regulator. And lastly, objective practice session 9, followed by dealings on economic botany and plant biotechnology in detail, after which we will be solving CUET model papers 3 to 5 sets. Okay, 3 to 5 sets of CUET model papers will be solving. So this is the complete structured plan for 60 days. Make use of it and this will be very useful. This is coming, the plan is executed from tomorrow only, okay, and this is accessible to all the students in this YouTube platform, okay. It is accessible to all the students who are going to appear for CUET PG Botany 2026, which is very useful for you, fine, right, because this is accessible to all, for all the students in this public platform. Make use of it. And if you are still having any doubts with respect to this exam CUET PG Botany, you can drop a message in the chat box. I will be addressing all your doubts, okay. And here in Narayangooda, Hyderabad, in our institute, you will be getting the live offline sessions, okay, live offline sessions where you can come and listen the classes, practice the objective with the mentors and tutors, okay. So make use of this public platform. If you are here and near, you can come to the institute and access the classes as well in offline, fine, right. So that's all for today's session. I wish you good luck and all the best for your exam, and I am with your preparation. Let's make a grand hit from tomorrow, okay. Yeah, thank you, ma.